So hopefully what I'm going to go through today, just go over, talk briefly about what is soil from my perspective, look at some different soil properties and the influence of those soil properties on soil carbon, look at physical, chemical, and biological properties that we might be measuring or should be considering when looking at soil, trying to discuss a bit about the impact of soil on, or the impact of climate change on soil, and the influence of some agricultural practices on soil health itself. And hopefully we'll get all that done within 50 minutes so you can ask questions at the end. So we have to understand that everything above ground is related to everything that's going on below ground. If we don't have healthy soil, we don't have healthy ecosystems growing above ground, and we as human beings require that in order to survive. So those things are very, very tightly tied together, and we need to support those three things going forward. So we look at the soil. In our lifetime, soil is generally considered a non-renewable resource. It takes a long time for it to form, about 1 to 2 centimeters, and about 400 years to develop a good soil horizon there. It's really important for terrestrial productivity, though. If you don't have a good, healthy soil, you're not going to have things growing above ground. But the soil itself is more than just organic matter, mineral material, and water and air spaces. So we look at soil formation. We're looking at 5 main factors that influence soil formation: the parent material, time, climate, organisms, and the topography. And those things influence how soil develops. There's a bunch of different processes that are involved within soil as well. We've got transformation of material and minerals in that system. We've got translocation. We've got materials moving up the soil profile, things washing down the soil profile. We've got material that's added to the topsoil, added to the parent material, causing soils to change. And we've got loss of material. We've got water that pushes minerals through, pushes clays through, changing the different soil profiles or soil horizons. So when we're looking at the soil itself, we're considering these 3 different groups of properties. We're looking at biological properties, chemical properties, and physical properties. We tend to do really well with the physical and chemical properties. It's very easy to go out and measure bulk density, nitrogen, phosphorus, potassium. We can send that to the lab, get it done. We can look at the stability of the soil. The one thing that we tend to neglect, because it seems to function, is the biology. So I'm going to focus more on that this morning, but we do have to consider the other 2 components as well, because the 3 of those things together have the soil functioning in a proper, healthy fashion for us. So we want to define a term. We're going to start defining a few terms. We're going to look at soil health. And this is a very large, broad term that somebody's come up with, and it covers a lot of territory. So we're looking at the ability of a soil to function as a vital living system, to sustain biological productivity, promote air and water quality, and maintain plant and animal and human health. That's huge. That leaves us wide open for total failure and makes it almost nonsensical as trying to prove something like that. But we persevere anyways, and we try to look at soil health as that system functioning below ground in a way to provide services for us. So when we're talking about a soil, a healthy soil, what is a healthy soil? Is that a healthy soil? If you're trying to grow cactus, that's awesome. If you want to grow strawberries, you're going to be challenged to do that. So you need to consider what you want your soil to do. Most of the ecosystems, the soils are going to function and provide the services needed for that ecosystem. From a farming perspective, we have to understand what it is we want to do with the soil, and we have to see if the soil can provide those services for us to perform. So we have to figure out ways to help the soil work in a manner that we can get the results that we want to get. So we need to look at different things to measure and different way to assess those things. Potentially, we're looking at a variety of different properties. Some of these properties are very dynamic, short-term things that change over time. So things like the biology below ground, very, very quick to change. Some organisms live from 2 to 3 days, some of them from 2 to 3 months, some of them for a couple of years. Very different from something like parent material or the topography of your farming area. That's really not going to change over 2 months, 5 months, 5 years, 50 years. that's pretty stable. So that's something that once you know what it is, it's really not something you want to monitor in the long term. We have to establish some sort of criteria. We have to figure out what we want to look at, and we have to establish that baseline information to monitor over time any changes in those soil ecosystems. So we want to look at assessing different chemical or physical properties over time and make sure that we're looking on a proper time scale to monitor those things. We have to establish baseline values. What's a good value for nitrogen or for phosphorus? What's a good bulk density to have in your soil? Those things we need to identify and establish and monitor those over time to see if their things are changing over time, if we're influencing them with the practices that we're performing on the land itself. And at the end of the day, we're not looking at single properties individually. We're not just assessing bulk density. We're not just assessing the nitrogen or the phosphorus content. We're not just looking at the microbial population. We're looking at all of this data together, and we need to integrate it in some fashion that will provide us a way of measuring and assessing the soil function or soil health. I kind of think of soil as a watch. So we're looking at it and we want to know the time. And as scientists, we've turned it over, we've looked at the back and we said, oh, look at all the gears. This is amazing. And as scientists, we've pulled it all apart and we started looking at all the screws and all the gears and the springs, and we've taken them off on our little side adventures and put them in little boxes, and we studied them like crazy, and we realized that that gear, that's so important. And what's happened is we've kind of forgotten how to put the watch back together. And at this point, the one thing I think we're missing overall is we got to find a watchmaker again. We need to find some way to put that watch back together, because right now, ladies and gentlemen, it's correct twice a day. And the rest of the time, it's a little off. So that's something we need to start looking towards. How do we start bringing together all of these different pieces of data that we are collecting over time and integrating it into making a complete and functional watch again? So some of the things we're going to look at, we look at physical properties. We're looking at things that we typically measure very easily these days. We're looking at bulk density. We're looking at soil texture. We look at erosion potential, the hydrology, the topography, aggregate stability. These things are very, very easy for us to measure, and we try to look at using those as some indication of what's happening below ground system. How do we go ahead and do that? Very easily, but what do we do with all this information afterwards? We have a pile of data here. Can we use just one of these items to give us some indication of what's happening below ground? Perhaps. Again, if we're trying to rebuild a watch, we cannot look at one gear or one spring. We need to figure out a way to start integrating all of this information together. If we start looking at soil chemical properties, we can look at soil organic matter. We can look at micronutrients, pH, salinity, the cation exchange capacity, the electrical conductivity, and we can generate information for these different parameters. But how do we use that information to tell us what's happening below ground? Can we use just one of those factors or pieces of data to tell us what's going on? Again, I would suggest, depending upon what you want your soil to do, maybe one of those components is enough. But we need to look at a way to integrate the chemical and the physical and the biological properties to give us a better picture of what's happening below ground. One of the things that I've been asked to talk more about today is soil organic matter, and how do we enhance the ability of our soils to develop that soil organic matter below ground, and how does that influence soil organic carbon and carbon sequestration. So soil organic matter is basically just plant and animal residues that are starting to build up on the ground. So the leaves fall to the ground, bird feathers hit the ground, cow manure hits the ground. All of that organic matter is showing up on the soil. And how does that get incorporated into the system? Does it spontaneously atomize and just release all the nutrients into the ground? It does not. It is actually influenced by soil organisms. It's that decomposition process of this organic matter as it hits the ground and breaks it down and immobilizes some of those nutrients and releases some of those nutrients and keeps the system functioning. So soil organic carbon is a part of soil organic matter. Soil organic matter is made up of a variety of nutrients and minerals, Soil organic carbon is one part of that. Usually about 50 to 60% of soil organic matter is your carbon within that system. And all of that soil organic carbon, as it's consumed by the microbes, by the fungi, by the springtails, by the mites, by the earthworms, that becomes part of their biomass. And that's stored within the soil system and slowly released over time as those organisms respire, as they consume other organisms, as they die, as they defecate in the soil. Now, the soil organic matter, or soil, contains about 3 times as much carbon as the atmosphere or terrestrial vegetation. So it becomes a really good sink for holding onto carbon. Now, how does that compare when we're looking at these other systems? So we're talking about vegetation, about 610 gigatons of carbon as a reserve. Atmosphere, about 600. The soil is about 1,500 or more in that top 2 meters of soil. Sediments and rocks, significantly more than what you find on the top soil. This is something that is in constant cycle within the system. It is not some sort of stagnant amount of carbon that sits there. It is constantly being consumed, decomposed, and recycled within the soil system itself. So when we're looking at measuring, how do we measure soil organic matter? How do we measure soil organic carbon? What models are in place, and how have we done it in the past, and what are we going to do in the future for assessing this? We can look at the classic humification process, and we've all heard about this, where organic matter decomposes, and as it breaks down, it leaves behind different amounts of humic or recalcitrant organic matter that doesn't break down in the system. I would suggest that guaranteed there is some bacteria that's going to break down that piece of carbon at some point, because it's going to be able to do that. So there's not going to be carbon that's left there not being able to decompose because we can't get it. We can't get something to decompose it. It's just that it can't be reached at that particular time, or there's a better source there. So from that classical humification process, we're moving into something that people are using called progressive decomposition. So we're looking at organic matter that is slowly broken down over time until a point where it's all gone and been absorbed into the ecosystem. And that's why we're constantly having to add organic matter back into that system, because it's being removed from the system. It's being immobilized in microbial biomass and other organisms' biomass. It's being released as available nitrogen and nutrients and carbon for the plants to take up so they can grow and produce whatever it is they're producing, so that we can harvest whatever it is we're going to harvest, or whatever the cows are going to feed, or the sheep are going to feed on. So that progressive decomposition goes into a new model that's come out. It's called a soil continuum model. And this is the constant addition of organic matter that's needed to the soil, because this organic matter is constantly being broken down and used to maintain a functioning soil ecosystem. So, wow, it looks so much better on a smaller screen. It's really washed out. The idea here is we're looking at plant and animal residues, and as they are being broken down, they're into larger polymers. They're into smaller polymers here, smaller, until it's just carbon that's left at the end being released into the environment. But each of these steps, we're looking at carbon that's being locked into the system. It's being retained in that system. It's not being released all at once. It is slowly being released over time. A lot of the models that are being used to give us a better understanding of soil organic matter and soil organic carbon tend to neglect the biological activity that's going on below ground. This becomes a critical component for looking at soil carbon and soil carbon sequestration. If we don't have the biology as part of the model, it becomes really difficult to get an idea of what's happening to the carbon below ground. Some newer models that are coming out of Russia are actually starting to include biological activity in that model development. So we're looking at faunal excrement. We're looking at the necromass, or the dead tissue that's falling into that soil system. And we're looking at earthworm activity. And that's something that really hasn't been considered for a lot of these models when we're looking at soil carbon below ground. Now, how can we assess carbon. What new ways, besides the old way where you just take a soil sample, you bake it in the oven at 500 degrees Celsius for 2 or 3 days, and you look at carbon loss. There are a variety of different fractions of carbon. There's labile carbon, there's more recalcitrant carbon, there are different size fractions of the carbon. How can we assess that to get an understanding of what's happening to the carbon in that soil system? We can look at something like thermal stability, so we can look at carbon breaking down at different temperatures and what portions are coming off at different times. We can look at using drones to fly over fields, and we can look at spectroscopy and use precision agriculture techniques to assess, using infrared spectrometry, what's happening to carbon sources below ground and how those map out over time. One of the big things about soil carbon is how important it is to soil health, and because so much carbon is used by the plants, so much carbon is used by the organisms, so much carbon is used by us, that needs to be constantly added to the system and replenished so the system can maintain that cycle that it's going through. So whether we're looking at a carbon cycle like this, nitrogen cycle like this, phosphorus, or sulfur, all of these actually go together in the below ground ecosystem. All of these nutrients are part of a larger system. Again, we're going back to this watch idea. Instead of just looking at individual springs or sprockets, we're trying to integrate this together. All of these things you'll see go through that black box. And the black box is the soil biota, the soil organisms below ground. Everything is going through that black box. And it's a great black box, but we have very little idea what's going on in there. It looks something like this. It's a very complex food web below ground. There are a variety of organisms there feeding on each other, breaking down organic matter, releasing nutrients, allowing decomposition to occur in new and different habitats. When we're looking at the biology below ground, we're looking at a variety of different groups. We're looking at the microflora, things like the bacteria and the fungi, the microfauna, things like nematodes and protozoa, the mesofauna, my favorite group, the mites, the springtails, and the macrofauna, things that are easily seen by us above ground, things like earthworms and beetles and isopods. Now, we not only have a lot of different species. In fact, in a single teaspoon of soil, you can have anywhere from 5 to 10,000 species of bacteria. In a square meter of soil, you can have anywhere from 200 to 300 species of arthropods. And just to put it into perspective, in the province of Alberta, there are 95 mammal species. 96 if you count Bigfoot crossing over the Rockies, right? So the below ground habitat is an amazing habitat, and I've really enjoyed studying it the last 20 years of work that I've been doing. It's a very interesting habitat. It's dark, it's opaque, and it's three-dimensional, which makes it very challenging to work in. But the diversity of species below ground rivals that of the coral reefs or tropical rainforests. In fact, some people have called it the poor man's tropical rainforest, which is good because I don't get a lot of research funding, so I can't afford to go to the tropics. I can just go to my backyard and sample these things. So again, we're looking at hundreds of species in a square meter. We're looking at tens of thousands of individuals in a square meter as well. So there's a lot of stuff going on below ground on these systems. What do these organisms do below ground? So we're looking at how they influence the decomposition process. We're looking at how they influence nutrient cycling and nutrient availability, and they also affect soil structure. How many people go out with the soils guys on their farm? Sometimes you go out. Do they do texturing of the soil? Yeah? And have you ever seen anyone do taste texturing? They put it in their mouths and they kind of run around and say, ooh, that's a sandy, loamy. Yeah, so for just the kids. Yeah, there you go, right? So one of the things you want to notice here is the amount of fecal material that shows up from the mesofauna and the earthworms, right? You'll actually get horizons that are made up of fecal material from most of these organisms. In fact, one of the people I used to work with, his famous phrase was, bug poop grows trees. And that's something to think about. If the soils guy says, hey, you should taste this, just say no politely and let him do his thing. So the macrofauna, we've got the macroflora, for the most part, the bacteria and the fungi do the majority of the heavy lifting for fragmenting and degrading the soil organic matter. They're immobilizing nutrients within their own biomass. As they consume that material, they're also mineralizing nutrients, releasing those things and making them available for the plants to take up in those systems. Most of them are very sloppy eaters, so they're releasing slimes and mucilages as they feed on this material, and that's binding the soils together. That's making those soil aggregates and building the soil structure below ground. The microfauna, things like the nematodes and the protozoa, are feeding on the microflora. A single protozoa can eat anywhere from 15 to 20,000 bacteria a day. So they're busy every day consuming stuff. They're fragmenting soil organic matter. They're regulating the populations of the microflora. They're altering the chemistry of that soil organic matter as they feed on it and change the soil, fragment the soil organic matter. And again, they tend to be very sloppy eaters. So the slimes and the mucilages and the debris that's cast off as they're feeding helps build soil structure below ground. The mesofauna, the springtails, the mites, those sorts of things, as they chew through the organic matter, feeding on the fungi and the bacteria, the protozoa and the nematodes, they're increasing the surface area of that, making it more available for the bacteria and the fungi to start breaking that material down. They're regulating those populations, and as they consume the organic matter, they're changing the chemical structure of that material as well, making it more amenable for other organisms to break the material down. And they're also depositing a lot of fecal material. They're busy. They're doing their thing, and they're building the soil structure below ground. Things like the macrofauna, we're looking at a lot of the earthworms, we're looking at ants, we're looking at isopods, fragmenting the organic matter, increasing surface area of the organic matter, allowing decomposition to happen in a very fast and efficient way, stimulating nutrient cycling and the availability of nutrients for plants to take up, providing a lot of fecal material in there. Again, soil structure, building that up. They're also redistributing a lot of material as well. Earthworms, as they move deeper into the soil profile, will bring up mineral soil to the surface. As part of that process, they'll bring organic matter deeper down into the soil profile, making those nutrients available to the microbes and the bacteria that are deeper in the soil profile. So you've got that mixing in there that's actually helping to develop soil tilth. So you've got all these organisms. What does that mean when you're thinking of 1,000 or 10,000 species of bacteria in a teaspoon of soil? It's kind of hard to put a number on it. So we'll start looking at biomass values, and we've seen some estimates of about 15 tons per hectare of soil biota in most undisturbed grassland system. Microflora make up about half of that, about 4 to 9 tons. Earthworm biomass, if the earthworm population is half decent, about 2.5 tons. So this 15 tons represents about 100 to 200 sheep per hectare. Oh, depths here. So most of the biota is probably in the top 30 to 40 centimeters. Somebody owes everybody a beer.
Organic beer?
Organic, sure. Organic beer. I'll take that. Yeah. So in the top 30 to 40 centimeters, that's where most of the biological activity is going. So that's where you've got this sort of biomass going on. In a disturbed system, in a farming system, you might have a quarter of this at any one time with conventional practices. So it's definitely changing that system and how that system functions in the short term. Can we change that in the long term? We'll proceed and see what we can get to. How many people have seen this? Was it this year, last year? The Soil Your Undies movement? Yay. Right? So here they're talking about using this to assess the health of your soil by looking at the biological activity. So really, we're trying to break down white cotton carbon in the soil, and you're hoping that it breaks down. Do we know what's happening to that material? Something's breaking it down. Do we know what? When you've got 5,000 to 10,000 species of bacteria in a teaspoon of soil, I have no clue what species is doing that. But I'll bet you dollars to donuts that it takes most of them to do it. And we need to have a better handle on that community diversity and what's going on below ground in order to make sure this is happening. So we're talking about soil carbon sequestration here. What are we talking about? We're talking about the removal and long-term storage of carbon from the atmosphere. This can be done in oceans, in forests or soils, and it's usually through physical or biological activity. I'm going to focus on the biological activity, right? We're looking at decomposition of organic matter broken down by the biology. So the bacteria, the fungi are feeding on that organic matter. They're being fed upon by nematodes, by protozoa. They're being fed upon by mites and springtails. They're being fed upon by earthworms and the other macrofauna. All of those things are breaking down that material and incorporating that carbon into their own biomass, and they're retaining it in that system. So the bigger the population, the more carbon is going to be held within that system. And we're going back and thinking about the biomass. That's what we're looking to build up to retain carbon in the system, to have that healthy functioning ecosystem below ground, that diverse community structure with the population and the biomass there to retain the carbon in the system. Now, does it stay there for the long term? No, because eventually things die. And that material gets released back into the system. As it decomposes over time, things breathe, things respire, they feed on other things, and you're releasing carbon dioxide back into the system. But it happens at a slow rate. It's not released all at once, it happens over a period of time. And it happens kind of in a nice little order, right? So during the summer, things are really active, breathing a lot, and they're releasing carbon dioxide because they're doing what they do. In the wintertime, they slow down quite a bit. It's not to say that they stop. Some of them go to sleep, some of them don't. They're active at a very, very low rate. So they're still respiring, they're releasing very little carbon dioxide. But most of the biomass is retained below ground. So we're looking at trying to enhance that community structure to retain the carbon in the long term below ground and slowly release it back over time. So what influence do agricultural practices have on this below ground community, this below ground ecosystem and its functionality? We can look at a variety of different practices. We can look at the influence of crop types and rotation. We can look at different soils and the influence that has on the organisms there. Cover crops, different tillage practices, grazing, fertilizers, pesticides, organic amendments, and that's something that we see a lot more of now. We've got a lot of organic matter that's being added to farming systems, whether that's manure, whether it's straw or bedding material, whether it's compost tea or some form of that material. We've also got more permanent plant growth that's being considered. We can actually look at some of these modifications and trying to offset potentially as much as 20% of our carbon CO2 emissions annually with using some of these modified systems. As an example, some work I did down in southern Alberta looking at native grasslands, and this is just the density of soil organisms in these systems. Density of mostly the mesofauna, so springtails and mites. Here we're looking in this system about 350,000 individuals per square meter in native soil. And material that was turned into grassland, we're looking at about 50,000 individuals per square meter. In older grassland, this is a system that had been left about 25 to 30 years, and we're just below 200,000 individuals per square meter here. So we can see in disturbed systems, you've got this generally a reduced diversity and density of organisms there, so you're going to have very limited capacity to store carbon in that system, a very limited capacity for that system to function properly or at an optimum level. When we're looking at trying to reduce carbon emissions, or we're trying to sequester more carbon in these systems, we can look at two different types of groupings for most of this stuff. We're looking at reducing or displacing emissions, so increased efficiency in farming machinery and practices to reduce the amount of fuel that we're going to use, or we can look at enhancing carbon renewal. So we're going to try to interrupt that natural system and the addition of material and trying to keep that locked below ground instead of having it cycle through in a normal way. Most conventional systems most conventional agricultural systems, we're looking at a homogeneous soil community that's there. It's something that's kept in a very sort of a very, very simple state, a very, very primary succession sort of stage. So you've got this constant disturbance that's happening to that system, so the community never has a chance to be developed fully, become very diverse. It would be kind of like going down to Calgary, and nothing against Calgary, even though I'm from Edmonton, but if you went to 17th Street and you sort of bulldozed it and piled it up at the end of the street, and then you spread it back out the next year, you're not going to say that that's 17th Street and it's working really well. Right? It takes a while for it to recover, but if you're doing that every year, what's going to happen to 17th Street? People aren't going to come back to that, right? So we want to try to look at not impacting the system as heavily as we can. We want to try to maintain a certain level of non-disturbance and allow those organisms to come back and function in a normal way. With tillage practices, we're looking at a constant influence or constant change to physical and chemical properties in the soil. We're losing soil structure for the most part. We're increasing the risk of soil erosion. We're losing the soil organic matter. And the reason we're losing soil organic matter is because we're incorporating the soil organic matter in, making it available for the bacteria and the fungi to decompose it so much more quickly, and then it's gone. We're not looking at building up the capacity to retain that soil organic matter and have it break down slowly over the longer term. As a result of that change in habitat and the chemical properties, we're losing the soil biota. We're creating a reduced community and its function. In fact, most of what happens when we're looking at tillage and just conventional tillage, we're increasing the bacteria. It's becoming a bacterial-dominated community, and we're influencing the type of organisms that are going to feed on the bacteria, limiting its capacity to break material down and cycle nutrients and make them available for plants to use. Using something like no-till or zero-till practices, we're looking at trying to increase the structure of that soil, making it more amenable to organisms and their habitat there. We're looking at trying to immobilize more nutrients within that system and make that system function more properly without our influence or impact to it. We're looking at potentially reducing the decomposition rate. So we're looking at slowing things down, tying up nutrients within that soil ecosystem, retaining nutrients and carbon in that system in a longer term and releasing them more slowly and allowing plants to take up those nutrients when they need to take them up. We're retaining moisture, we're making the habitat better, we're allowing for an increase in density and diversity of the soil organisms that are living below ground. Grazing also has an impact on the below ground ecosystem. We're looking at, depending upon the number of animals that are grazing on a site, we're looking at increasing bulk density. We're going to change the soil temperature. We're going to make things warmer. We're going to allow decomposition to happen more rapidly, and we're going to lose nutrients and carbon in that system faster. We're going to lose soil organic matter. We're going to have higher erosion rates. We're going to have lower soil moisture. We're going to change the aggregate stability of that system, and we're going to make it less hospitable for the organisms below ground with conventional grazing. So we need to pay attention to the number of animals that are grazing in those ecosystems. The addition of fertilizers and pesticides become a problem. The more of this material we have to add, we can have direct effects and indirect effects on the organisms below ground. Direct effects, we're altering the chemistry of that below ground habitat. Also, the organisms, depending upon if they come in direct contact with that material, they're going to die straight away. Or we can have indirect effects to that ecosystem. We can change the chemistry, we can change the food availability, we're changing the habitat or the climate of that system. We're making it almost impossible for some of those organisms to live in that habitat. The addition of nitrogen fertilizers, we're looking at changing or increasing the osmotic pressure, so it makes it harder for organisms to live in there. We're changing the soil pH, we're changing the amount of soil organic matter in those systems. We're changing the food supply potentially for the animals living below ground. That's going to make it hard for them to live there. We're going to reduce the density, and we're going to make that system function more poorly. The addition of urea fertilizers, we're looking at changing the community structure again, more bacterial dominated system, bacterial feeding nematodes on that system. The addition of pneumonia, we're going to change the bacterial community as well. In use of liming, we're going to change the earthworm abundance there. We're going to have more of earthworm densities increase as opposed to enchytraeids enchytraeid worms that are going to be reduced. Two different species of worms belonging to the same group, but functioning at a different level, right? Most earthworms, the lumbricids within Canada, are introduced species at this point. There are very few native species left. I think there's about 4 or 5 of them. Most of them are on the west coast in refugia. All of the species we're finding now are European ones that have been introduced as people came across, planted their favorite plants from back far east, and now we've got earthworms showing up. If you're going out and fishing somewhere, how many times have people gone out to fish and taken their worms and at the end of the day said, yeah, I'm not going to carry these back, and just sort of dropped them in the woods? And we're starting to see earthworms show up in very, very interesting places where they haven't been before. That's not to say that these systems aren't going to function well, it's just that they're going to function differently, and you have to be prepared for understanding what that's going to cause. Application of pesticides can have varying responses on the soil organisms below ground, and that's going to influence decomposition rates and the capacity for the system to function properly. Depending upon what's applied, how it's applied, and the organisms that are present, some species decrease in number, some of them increase, some of them are not affected at all, but it really depends on what's there beforehand. And we have very little idea of the density and diversity of most of the below ground ecosystems. Overall, what practices enhance soil health and which practices reduce soil health? For most of these, we're looking at no-till or reduced-till that's going to make the soils function a little better. The use of cover crops, diverse crop rotation, perennial crops, crop residue retention, organic fertilizer application, and these are things that the previous group were talking about doing on their farms, and a lot of people are doing right now as well. We tend not to see a lot of tillage happening these days. We're tending to see less fallow fields. Annual cropping is something that we're not doing a lot of. A lot of people are looking at changing from inorganic fertilizers to organic fertilizers to help those systems function better and to reduce the impact on the biodiversity and things that's happening below ground. There are a couple of things going on between us and between nature. We've kind of become slaves to the soil. We actually have to go in and we have to prepare the seed beds, and we have to do the planting. We have to provide chemical pesticides and herbicides to control crops. Thank you. And we're applying chemical fertilizers because we want to have a certain yield. But I think, oh, darn it, nature does that, a lot of that stuff already, and does it for free. Right? A lot of natural seed dispersal, a lot of natural control of weeds and insect pests and pathogens, assuming that the biology is there to provide those services, and a lot of decomposition and nutrient cycling of organic matter, provided that organic matter is there for those organisms to use. So we can either do a lot of this work ourselves, or we can try to harness the organisms below ground and have them help us out to do the work we want to do. There's a variety of studies that have gone on in the past. Alberta, I can't remember what they were called at the time. I think it was Environment and Agriculture, but now it's Environment and Parks or something. They ended up doing a 10-year study looking at soil quality within the province. And they've got a bunch of benchmark sites that they've placed throughout the province, and they were all sampled about 10 years ago. They've got this material that's been archived, so we're looking at an opportunity here to go out and try to assess what's happened in the intervening 10 years and use that as an idea of how various practices on these farming sites have influenced the soil health or soil quality. We can use that as a benchmark, and we can go forward and look at soil chemical and physical properties, but we can also add the soil biology to it as well and get a better understanding of what's happening in those below ground systems. So again, what we want to try to do is we want to try to get healthy soil that's going to provide food, that's going to help reduce climate change, and it's going to help us retain water within that system so that we can build that watch again and use that to help us tell us what's going on in the below ground system. And so we can just take a look and know that we're dealing with a healthy, happy soil. So yeah, all this above ground diversity, terrestrial productivity, the key to it is the below ground soil and the healthy functioning of that soil system. Thank you very much for your attention.